Okay guys, welcome to this next video. In this video, we will be focusing on DNA and protein synthesis and all the BMAT section 2 questions regarding this topic. We have now dealt with the beastly topic of inheritance, guys, and that's really the hardest thing you will see in the BMAT section 2 biology. This topic is pretty straightforward, but we're going to go through the key concepts as we go through the questions, guys. So let's go through it. So BMAT 2019, question 21 is the first of these. So it says, which of the following statement is or are correct for a healthy human cell? So what I want you to do, guys, is pause the video, give it a minute, give it a go. I don't want to go through each of the individual points now because I would obviously be giving the question away. But if you're unsure, don't worry, guys. Just carry on watching and we'll go through the solution. But if you can, guys, give yourself a minute and give it a go. Okay, welcome back, guys. So the first one, it says a protein section coded for by part of a gene consists of 500 base pairs, right? Will have a maximum of 166 amino acids, right? So remember, guys, a gene is just a section of DNA which is obviously going to have base pairs because that's the essentially the monomers of DNA. What they're referring to here, guys, is they want you to recall the idea of the triplet code. So each amino acid is coded for by three bases. So if each amino acid is coded for by three bases, the amount of bases you would need to code for 166 amino acids will be 166 times 3. So if I just do quick column method, it's going to be 18, 180, and then 300. That's going to be 8, 9. Yeah, so you're going to need 498 bases to code for 166 amino acids. But this one has 500 base pairs, so it has two more. But remember, those two, they basically won't do anything because you need at least three bases to code for amino acid. So this one, 500 base pairs, would actually give 166 amino acids. The two base pairs that are left over can't do anything because of the triplet code, obviously. So each amino acid is three bases. So the next statement, guys, says a liver cell and a mature red blood cell from the same person will both have a chromosome number of 46, right? Actually, I forgot to add the whether one is correct or not. And yeah, so point number one, that is going to be correct. And the next one, a liver cell and a mature red blood cell from the same person will both have a chromosome number of 46. So I think we went through this before. The diploid number 2n is 46, making the haploid number in gametes 23. So the diploid number, remember, guys, occurs in any body cell, right? So a liver cell is obviously a body cell, therefore the chromosome number will be diploid and it'll be 46 in humans. But here's a trick, guys. Remember, you need to know basically what is a mature red blood cell. Remember, a mature red blood cell is just a bag of hemoglobin, and that means that it doesn't have any nuclear DNA of any sort in its interior. So that wouldn't have any chromosome number at all. So this would be incorrect then. And then the last point says the number of adenine bases in an allele must be the same as the number of guanine bases. And what this is referring to, guys, is your idea of base pairing. So obviously you have A to T going with two hydrogen bonds and C to G going with three hydrogen bonds. So that means essentially what that means is in an allele, the number of A would be the same as the number of T because they pair up, and the C would be equal to the number of G because they pair up. So adenine and guanine, they can be different because these two don't pair up. So this is going to be incorrect. Therefore, guys, the answer, I hope you got this, guys, was B. But if you didn't get it, no worries, guys. The whole process is about learning, and hopefully we sort of cleared all the things you're unclear about in this question. And I hope to see you in the next one where we can learn more. Okay, guys, so in the previous question, BMAT 2019, question 21, we took this question where we looked at some ideas of, the essential elements of, DNA, the idea that it's a triplet code and the idea of base pairing and just how DNA works in general. So we're going to take that a step further, I hope, with the next question. And the next question, guys, is BMAT 2017, question 5. So it says SCID, severely compromised immunodeficiency. That's essentially what it stands for, is an inherited condition in humans. People with some types of SID are unable to make a functional enzyme necessary for the production of healthy white blood cells. And obviously, white blood cells are essentially the key cells you need to fight off an infection. It says a scientist studying these types of SCID compare features of the DNA and the structure of enzymes in people who have the condition and people who don't have the condition. And it says, which of the following features may have been different? So pause the question, guys, give this a go, and be sure to come back after a minute where we'll be going through the word solution. Okay, guys, so let's take a look at this. So it says the allele of the gene. Remember, allele is just the version of the gene, right? So for example, the gene here is going to be the enzyme. The allele can be functional enzyme or non-functional enzyme. So the allele of the gene um, coding for the enzyme. So can that be different? So basically, what let's just quickly go back, guys, and see what this is. So people are unable to make a functional enzyme. So that's basically the difference. So people who don't have the condition can make the functional enzyme, whereas people who have the condition cannot make the functional enzyme. And then obviously, the allele is where the enzymes are proteins, and proteins come from genes, and alleles are a part of genes, right? So, so let's just actually write that down. So basically, you have a gene, or essentially an allele, which makes essentially a protein via the process of transcription and translation, and then the protein is going to make an enzyme, right? Because enzymes are proteins, right? And basically, these people don't have this. They don't have this enzyme. So essentially, anything along this path can be messed up, essentially, so that they don't have the enzyme. So the allele of the gene coding for the enzyme, yes, so that can be different. So perhaps they can have a mutation in that allele, which means, for example, you get a frame shift and therefore you get a truncated protein. That's obviously going to be called a nonsense mutation, and therefore you don't make the protein, and therefore you don't make the enzyme. So this can definitely be the case, right? The order of amino acids in the enzyme. Okay, this can also be the case, right, guys? So what you can have is a subtle mutation, which is called a missense mutation, right? Meaning that you're still going to transcribe and translate your protein. You're going to get a full-length protein, unlike the previous one where you get a truncated protein, but maybe a critical residue. So this is an enzyme, isn't it, guys? So this is the normal enzyme, and let's just say you need, for example, amino acid X there and amino acid Y there to hold this in place, right? Maybe this enzyme, guys, with the missense mutation, it only has everything's the same, but the only thing is there's no amino acid X in there, and then obviously that can mean that that's going to mean that the active site won't fold up properly, and essentially the enzyme is no longer functional. So the order of amino acids, yes, that's going to have a huge, it's going to be critical for the functioning of the enzyme. So this can also be different. So the next one, the order of the bases in the gene. Um, Coding for the enzyme. Obviously, this can be the case. So again, you can have a subtle, addition of three bases or deletion of three bases. Remember, that doesn't cause a frame shift, guys. But that's obviously going to change the ordering of your bases in your gene, and then that's going to mean your protein is going to be different, and therefore your enzyme is going to be different. And perhaps even this could mean that the protein is completely truncated, as the case was in the first one. So this can definitely be the case. And the shape of the active site of the enzyme, of course, that's a critical part, which is going to be responsible for the reaction. And maybe the reason they might have the enzyme, but the enzyme might not be functional because the shape of the active site, as we can see on the right, is not right. So this can also be the case. So basically, any of these can be the case, guys, and therefore the correct answer would actually be G. So um, hopefully, guys, you've managed to get that one right. And if not, hopefully my explanation has made sense to you all. But I hope to see you in the next question. Okay, guys, so the previous question, BMAT 2017, question 5, we were looking at this inherited condition and different factors which might have led to the enzyme in the inherited condition from being non-functional. And hopefully that has made sense, guys. And as always, guys, if it didn't make sense, just comment down below and I can give you perhaps a more tailored solution to you. But now, guys, we're going to go on to the next one, which was BMAT 2011, question 21. All right, so it says using the table, select the correct answer from the table. So give yourself a minute, guys. Pause the video now. Give it a go, and we'll be back after a minute where we'll be going through the question together. Okay, guys, so let's look at it. So what we have here, guys, we have three different cells, P, Q, and R, and they have different quantities, relative quantities of nuclear DNA. So Q has double P, and R has nothing, right? So essentially, guys, for the BMAT, the only cell that has no nuclear DNA, and as far as I can think now, the only cell that has no nuclear DNA is essentially red blood cells, right? Because remember, guys, red blood cells are just a bag of hemoglobin. They don't want to have any internal organelles. They don't want to have any nucleus or DNA because they want to pack in as much hemoglobin as possible to maximize the efficiency of oxygen carriage. So already, you guys, we know that R is going to be a red blood cell, or another possibility they give here. So it could be this, right? Or another possibility that can obviously happen, which isn't so natural, is an enucleated egg cell. right? I think we looked at a question before in one of the previous videos where we were looking at Dolly the sheep, and essentially this is one of the processes that happen when you make essentially clones like Dolly the sheep. So you obviously take out the nucleus from the egg, and then essentially what you get is the nucleus from a diploid body cell, and then you fuse those two together. So yeah, this is a like sort of more artificial way of having a cell which has no nuclear DNA. So that one can obviously be the case. And then let's go with P next. So P has only half the amount of nuclear DNA compared to Q. So basically, what's going on here, guys, is that P would basically be the N and Q would be the 2N. The N being the haploid, and the haploid number of chromosomes is essentially found in gametes, right? Whereas the diploid number of chromosomes, we have double the amount of nuclear DNA, is essentially found in any body cells. So any of the body cells. So we know then that P is essentially going to be a gamete. So you can have gamete, obviously, and gamete itself is an umbrella term for sperm and egg. So you can also have one of these guys. And then Q is just any body cell. So a cheek cell would be fine because that's part of your body. Adult stem cell, that would be fine. For example, you have stem cells in your bone marrow that produce the various different types of blood cells. Nerve cell would be fine, and a fertilized egg cell. By this point, it would also be diploid and therefore considered a body cell. So the one that overlaps all three is going to be D, and therefore D is going to be the correct answer. So hopefully that has made sense, guys. And I believe this topic was really short, actually. I believe, yeah, this is the last question for the topic of DNA and protein synthesis. Not really that difficult, guys, as you can see. It's just applying your basic principles of how DNA and protein synthesis work together in a concerted manner. And the next video, guys, will be going over, as you can see, the topic of genetic engineering. I know a lot of students get confused by that topic because it's not really covered at AS and you covered it at GCSE. So it's like in your memory 1 year back, but we'll be sure, guys, to go over it properly. But other than that, guys, hopefully that was useful, and I look forward to seeing you in the genetic engineering video. So see you guys soon.